Hi everybody, my name is Noah. I'm from the University of Maryland. Today I'll be talking about some reservoir computing experiments on neural networks that we're doing to investigate neuroplasticity. I guess I can just get started. Give us extra time. Yeah.
Good.
Okay. So just an outline of what I'll be talking about. First, what reservoir computing is, and then also how we can do that on living neural networks, as well as the goal, which is to observe plasticity, study plasticity in neural networks with that reservoir computing framework on a more network scale. Yeah. Okay. The outline of what is a reservoir computing. First, a reservoir computer is a subset of the recurrent neural networks, and the key difference of these recurrent neural networks. Oh, sorry. I didn't realize this. My bad. That's why I need extra time.
It's attached to this thing.
Yeah, I'll stay around here. Yeah, that's fine. Thank you. Anyways, what was I saying? Yeah, reservoir computing is a subset of the recurrent neural networks where you'll see there's loops in your network rather than a feed-forward neural network, which will be a function and information would flow straight in one direction. So here, what that means is that our network is a dynamical system. You'll have memory within it, so as information flows, it'll revisit certain nodes and such. So that's one key feature of the reservoir computer. Another is this randomly assigned and static reservoir adjacency matrix. So what this means is that this whole reservoir layer is random, and we don't have to tune any of these parameters or even the network structure at first, and it will stay as it is throughout the entire learning process. And yeah, one other important feature of the reservoir computing is that the training is done differently from a typical neural network learning process, whereas on one hand, you usually do the gradient descent. Maybe you'll have to train all these connections. In reservoir computing, you go straight from all the reservoir activity straight to the output layer. And what you do is you do a regression in order to obtain this set of output weights. And this is really good because linear regression is very easy. You don't have to do gradient descent. And when you're doing gradient descent in a recurrent neural network, you can have bifurcation problems when you're trying to go back in time. You have to do training throughout all of that, whereas this linear regression is just a simple matrix equation here. To talk about it a little more, the kind of regression that we do here, if you're familiar with linear regression, has this additional term, lambda, where we have a regularization term so that we want to give at least every node a chance to contribute to the information processing. So that's just one little difference between typical linear regression. But yeah, now that we know what is the reservoir computing, the experimental question is how we can implement that in living neural networks. In our case, we have a co-culture of neurons and astrocytes, but you will be only seeing the activity of the neurons here. So it's maybe the first is self-explanatory. The nodes will be the neurons. So here we can see that the neurons will be serving as the reservoir layer. More interestingly, you will need some way to input whatever training data that we want. And in this case, we're using these ion channels which live on the cell membrane. And what we can do is we can stimulate the ion channel with photons using some machineries that we have, and we can trigger action potentials at the cellular level. So cell by cell action potential, we can control over that using these kind of genetically encoded ion channels. Next thing that we need is to have a way to read out the response from the reservoir layer, and in this case we're using genetically encoded calcium activity, JGCamp, and this will allow us to read, as you can see here, an example video of the calcium signal within the entire population. And we can do like a cell segmentation to get individual cell traces to represent the electrical activity within the neurons. Yeah, so this kind of allows us to do RC in general on our biological neural networks. So one key difference between these living neurons versus the typical reservoir computing is plasticity, and this is the key thing that we want to investigate. If you remember earlier, I said reservoir computing has the static and random structure, And this is true at the beginning. When we plate the neurons, they're static and they're random. But over time, there are mechanisms by which this structure changes. So I'm going to talk about that a little bit. So the theory is that an adaptive dynamical network has some time-dependent adjacency matrix as a function of dynamical state or history. You can see here in this diagram that as the dynamics flows and information goes through the network, there will be changes to the topology, which then affects dynamics. So it goes back and forth like this. So that's a good framework to think about it. Just one example of this kind of effect, we're going to talk about the spike timing dependent plasticity. This is not the only or nearly the only effect that can modulate synaptic weights, but it's just one example of a well-studied one to show that there are many ways that the synapses change between the different neurons. So the way it works, this is an example of Hebbian learning, also known as cells that fire together, wire together. This one difference is, or example of it is that the timing between the firing of the presynaptic and postsynaptic cell, that timing will affect the synaptic strength. So here, for example, if you had one cell that fired right before another, then you would get an increase in the synaptic strength between those two. And as I mentioned, STDP alone is not enough to get a stable learning network. You'll have, you'll have strong links that just go stronger, and this will be unstable. So we know that in real learning environments, there are much higher order interactions between 2-3 cells or maybe other cell types that mediate this kind of learning, but it is an example of one of these learning mechanisms. So just to show that there is evidence for adaptation occurring in general, plasticity in general, which I'm sure we all know. Great. So now that we have reservoir computing on our neural networks, as well as this adaptation mechanism or plasticity mechanism that we talk about, we want to talk about how is information or dynamical information, embedded within the neurons. So just an example, this is the kind of training data example that we'll be using, such as Lorentz system. So the first question is a control experiment. How can spontaneous activity contain dynamical information? And to do that, you do that regression that we talked about earlier, where you will observe the spontaneous activity, in this case, of our reservoir layer, do a regression like this against the training data, and note that we have not actually input anything, so it is purely spontaneous. And you'll observe that there is some distribution of weights here, where certain nodes close to 0 are not participating, so that maybe shows that that cell is not involved in the information representation, whereas certain nodes or certain cells are very highly represented by their weights. And so what you expect to see, and which is what we have seen here, is that as we observe the spontaneous activity, this weights distribution doesn't really change. And that shows that there's no learning occurring in it. It can represent information arbitrarily because it is a dynamical network, but it's not doing a necessarily good job or a better job over time. And that leads us to the question that we want to study, which is plasticity. And in these experiments, you'll see now that we are actually inputting this training data. And the way we do that is we segregate the cells into 3 groups for each spatial dimension of the Lorentz system. And then we use that photostimulation that we talked about earlier to input this data. And you'll get output traces. This is just a representative of maybe the 3 different kinds, but you'll get output calcium activity. And then we do the same regression that we did earlier. But instead of just doing it once, we'll do multiple training periods. And the goal is to see, did that distribution of weights change over time? So that's our hypothesis, that information encoded by fewer neurons would imply that the network state adapted to the training data. So as we're actually inputting this into the system, does that, sorry, I'll just go back and show this. Yeah. Does this distribution change over time to be a more efficient representation of the training data that we put in? So maybe only a couple nodes would be very heavily involved, whereas during the control, you'd have a lot of nodes being necessarily involved. So that is what we're working on now. And I think with that, I will conclude just to say we're doing reservoir computing experiments on living neural networks with the goal of observing plasticity at the network scale. I'll thank my advisor and also my team for helping with all of that. Yeah, thank you. Any questions I can answer?
So with that, we can take questions from the audience.
Yeah.
How do you choose or train the connections from the input layer to the RC layer?
Yeah, that's a good point. So here you can actually see we just literally have a block diagonal matrix where you just put one into the, and there's no mixing terms. You could have that. I haven't even thought, I mean, I have thought about it, but I haven't really done it. It seems a bit arbitrary. You could, yeah.
Are the neurons picked randomly?
Yeah, it is important that we want spatial segregation. So we don't want it all to be in one, not segregation, we want distribution so that it's not localized each spatial dimension. But yeah, that's a good question. So I guess one future thing you could do is have those mixing terms so that there is integration of both of the dimensions. That could be interesting. But yeah. Mm-hmm.